Looking back at the past, I worry sometimes because sometimes you'll present old information, but it's foundational information, whether it's the biology of the issue that you're looking at or previous experience with an issue that previous experience has validity and applicability to the current situation that we're in.
Welcome to the BarleyBin. SaskBarley's goal is to take you right into the barley bin, not a hopper bin or a bin with in-floor augers, but the kind you'd spend a bit of time shoveling, getting itchy. It's worth it for the pint or steak afterwards, or a golden nugget you sometimes find uncovering itchy topics.
I'm Mitchell Japp, and today we're picking up a thread we started back in Season 1, Fusarium Head Blight. If you caught that episode, you know the basics. Fusarium Head Blight, or FHB, or scab, is a fungal disease that attacks barley and wheat heads. It's caused by a fungus called Fusarium. There are several species, but one we hear a lot about because it is so aggressive is Fusarium graminearum. When conditions are right, warm, wet weather, right around head emergence and flowering, it can be devastating. It remains hidden until it is too late. In barley, the main problem is that Fusarium can produce a mycotoxin called deoxynivalenol. Most people know it as DON, or by its less scientific name, vomitoxin. That name tells you everything you need to know. It's the disease barley farmers fear the most, and for good reason. It is Canada's most economically damaging cereal disease, year in and year out. So what's changed since we last talked? Quite a bit, actually. The science has kept moving. Today we're going to hear about the fundamentals of FHB risk that every producer should know, new research on spray timing that gives growers more flexibility in the field, a project looking at mycotoxins you may never have heard of, but that could matter a lot to your markets, and some genuinely novel genetics work that's finding FHB resistance in places nobody thought to look before. We're joined today by three researchers whose work is funded in part by SaskBarley: Dr. Kelly Turkington of Agriculture and Agri-Food Canada in Lacombe, Alberta, Dr. Randy Kutcher from the University of Saskatchewan, and Dr. Gurcharn Brar, now at the University of Alberta. Let's get into it. Before we get into the research updates, let's start with the fundamentals. Kelly Turkington has spent decades studying FHB across the prairies, in producer fields, in plots, and in collaboration with researchers across multiple provinces. When it comes to what actually drives disease risk in any given year, he puts it simply.
Really, the key factors for disease risk are the weather. That's the mother of all risk factors. The variety resistance that you have, and then the rotation that you're following. Those three factors have the biggest role to play in terms of disease risk. So we see that from research, but you can also look at farmer experiences.
Weather, variety, rotation. Three factors in that order. Weather is the one you can't control, but you can watch it. Warm temperatures and high humidity during the heading period are the conditions FHB needs to get established. Disease forecasting tools exist precisely to track those conditions and give growers a heads up when risk is elevated. Variety is your first line of defense before a seed goes into the ground. No barley variety is fully resistant to FHB and certainly not immune, but some are significantly less susceptible than others. Checking the disease resistance ratings in the SAS Seed Guide before you decide what to grow is one of the highest value decisions you can make for FHB management. Fusarium is an unforgiving disease. You can do everything right and still lose the battle with Fusarium. In the early 2000s, growers in northeastern Saskatchewan, the Melfort, Tisdale, and Carrot River areas were following the textbook on FHB management. They were doing what the experts told them to do, and they were still losing. Here's Kelly on what that looked like.
I'm a Saskatchewan boy, so this is a soft part in my heart for Saskatchewan, and the challenges in the eastern part of the province, especially northeastern area, Melfort, Tisdale, Carrot River area, I think. And growers were doing everything according to what the experts were telling them, and an approach coming out of North Dakota and people like Marcia McMullen in the early 2000s to look at using the most resistant variety possible, which for me it's reduced susceptibility. That's a whole other ball of wax. And then crop rotation, not planting wheat on wheat or barley on barley. And then, of course, spraying a fungicide. And even though they were utilizing those three strategies, and there are reasons for this, but they were still getting hammered with downgrading due to FDK in the harvested grain, especially for wheat. But more importantly, both for feed barley, if you're looking at the hog sector, or malting barley, the food sector, you had issues with dawn. And so frustrating. It's a difficult disease to manage.
Fusarium damaged kernels, FDK, is exactly what it sounds like. Kernels that have been visibly damaged by Fusarium infection. They're shrunken, shriveled, often with a pink or white chalky appearance. They're measured as a percentage of the sample and are a direct grading factor for both feed and malt barley. FDK is more common in wheat than barley because barley flowers while in the boot when infections occur. But high FDK does mean discounts. Combined with elevated dawn, it can mean outright rejection. What Kelly is describing was a real production crisis. Growers weren't being reckless. They were following the recommendations of the day. The problem was the recommendations weren't quite right yet, specifically on fungicide timing. That's what the next round of research set out to fix. It's important to note two key takeaways from Kelly's long career that have helped in the fight against FHB.
Just to take a minute to look back at your career, I'm gonna share what my couple takeaways are, and it's just two of many. But
sure.
The ones that stand out for me are no tank mixing fungicides with your herbicide. That was a good rule of thumb out of some of your research, and that wider spraying window for Fusarium Head Blight. To not get in there where the label says, as soon as you got 50% head emergence, you want all those heads emerged on your main stem, because anything still in the boot, it's not getting protected. So those were kind of the two takeaways that I came up with as very critical. Boots on the ground, easily applied in the field right now, results out of your research.
Well, certainly you've mentioned two that are very important, and the herbicide timing, lots of interest in that in the late 90s, early 2000s, and then over the last 20 years or so. We did some work at Melfort and Beaver Lodge and Lacombe in 99 and 2000.
The short version? Kelly's research was definitive. Adding a fungicide to your herbicide pass adds no value for leaf disease protection. It doesn't work, it costs money, and it increases the risk of building fungicide resistance. We're more familiar with herbicide resistance in weeds than with fungicide resistance, but it is a risk and one worth being wary of. Many of the fungicide groups we're spraying for leaf disease overlap with FHB suppression. Now, spray timing. This one is important because getting it wrong in either direction costs you. Too early and the heads you're trying to protect aren't emerged yet. Too late and the window for infection is already passed. The fungicide label for FHB has traditionally pointed growers towards 50% head emergence as the application trigger. Get your product on as soon as those heads are showing. Starting fungicide application that early can help with logistics if you've got a lot of acres to spray, but research out of North Dakota and Kelly's own trials in Alberta and Saskatchewan showed that the effective window both starts and ends later than the label implies. It's important to remember to stay within the label limits, but knowing the best timing within the label limits is valuable. Strong research like this may be a starting point to adjusting label limits, which would be a big help in effective and logistical management of FHB. Here's Kelly.
We didn't focus on the start of the label window because the information out of the States was dictating that that's not a good timing. So we wanted to look at what was considered to be the ideal timing at the time and then pushing that. And really in our research, seeing this and then looking at work that Gurcharn Singh did with Randy Kutcher and Durham Wheat, and then all of the work out of the States, various colleagues there, all indicating that that window extends much later than we first thought. So that provides a bit of opportunity for growers in terms of that's a pretty stressful time, and trying to hit that 3 to 4 days after head emergence in wheat, or maybe complete head emergence 1 to 2 days after that in barley. That's a pretty small window of application, and in a busy growing season, and then dealing with weather systems and so on. So I think we've got a good foundation of research, and hopefully we'll see some continued work looking at tweaking that and utilizing the disease forecasting tools that we have, the weather-based maps, and utilizing that to try and tweak when we need to go in.
That forecasting piece Kelly mentioned. There are weather-based risk tools available to growers and agronomists that track the conditions FHB needs to get established. PrairieFHB.ca offers a free risk mapping tool that combines temperature, humidity, and crop stage to assess FHB risk in Western Canadian cereal crops, because it's the conditions, not the calendar, that matter. It's a smarter trigger, and it pairs well with that focused window. Coming up, the disease produces more than one mycotoxin, and some of the others are ones the market is starting to pay attention to. When growers talk about the quality side of FHB, the conversation almost always comes back to DON, deoxynivalenol vomitoxin. Regardless of the name, it's the toxin that triggers downgrading, feed refusal in livestock, and market rejection. We measure it. We set thresholds for it. We've built most of our FHB management strategy around it. But DON isn't the only mycotoxin Fusarium produces. A mycotoxin, to define the term, is a chemically stable, poisonous compound produced by a fungus. Myco comes from the Greek word for fungus. And where there's Fusarium, there are potentially several of those mycotoxins. Dr. Randy Kutcher at the University of Saskatchewan is working with colleagues in the pharmacy department to identify and quantify the mycotoxins we may be under-measuring. Here's Randy on why that matters.
You've got a project right now about identifying mycotoxins in cereals. Mycotoxins, I think from a grower perspective, they hear about vomitoxin, DON, deoxynivalenol, and that's the primary one that we hear about. Maybe a little bit of ochratoxin A, but for the most part, it's DON. And so your project, it goes into a lot more different mycotoxins. Is that right?
Yeah, I mean, there's no shortage of mycotoxins, and some of these other ones are quite toxic compared to DON. My understanding is DON, things like animals like pigs, they just won't eat it, right? So it's feed refusal more than you're poisoning the animal, I understand. So DON is very important. It's the most common one. We've got good tests to test for it and quantify it, but there are many others. So there's like HT2, T2, nivalenol. There's NX3 and NX4, and you name it. There's quite a few. And then even with DON, there's what they call the masked mycotoxin. So it changes form in the animal or even in the plant. So it doesn't look like DON and you don't detect it, but then it can change back into DON because it's a very subtle change. Yeah. So Dr. Wong, who I'm working with, and colleagues over in pharmacy, because they have the equipment, we have a postdoc right now and a new graduate student who are looking at trying to develop methodology or tests using existing equipment like HPLC and mass spec to be able to identify and quantify these other toxins, so that if they do become a concern, because certainly in some countries like Europeans, I think, are much more aware of the toxins and a little more, stringent, if you will, or they're concerned that they may start restricting, grain coming into Europe with some of these other toxins, not just Dawn. So this project would address that.
And so these other toxins, that you're looking at in this, are they primarily Fusarium related?
Yeah, these ones are all Fusarium related. That's all we're working on.
A couple of terms worth unpacking there. HPLC, high performance liquid chromatography, and mass spectrometry are laboratory techniques used to separate and identify chemical compounds in a sample. They give researchers a way to go beyond saying, there's a toxin here, because these tools can identify, here's exactly which toxin or toxins, and here's how much of it. The masked mycotoxin piece is particularly worth noting. The idea that a toxin can disguise itself in the plant or the animal, registering as something harmless, and then revert to its original form is a newer area of concern. It means our current testing methods could be undercounting the actual toxin load in a sample. For now, Dawn remains the primary concern for Canadian markets, but this is the kind of research that positions the industry ahead of a problem rather than behind it. The work we've heard about so far is about managing the disease that's already in the field. What comes next is a different question entirely. What if the genetics to fight FHB are already sitting inside a variety we're already growing? Dr. Gurcharn Brar, formerly at the University of British Columbia, now leading the wheat breeding program at the University of Alberta, took an unusual approach to FHB resistance research. Rather than comparing different varieties against each other, he looked closely inside one, AAC Synergy. It's currently the most widely grown malt barley variety on the prairies. This project was funded by WGRF and SaskBarley. Here's how he introduced the concept.
So the whole concept of that work was um, in a genetically uniform variety, let's say AAC Synergy, you should not see any variation in any developmental or disease resistant traits or any traits, because the variety should be homozygous and each plant should be identical to the other plant. Like if there are 100 AAC Synergy plants, they should be all flowering at the same time. They should have same level of disease resistance, but that is not necessarily true. And that is because of different transcriptional programming of the genes that control some of these traits, and we call it, in a simple language, gene expression noise. So we try to quantify that and identify the genes that lead to these differential phenotypes among plants of a same variety. And we actually used AAC Synergy, and it is quite novel because it has never been used in cultivated crops to dissect disease resistance. So we were the first ones. Through that study, we actually identified some genes and associated markers which could be selected for or against to basically select a better version of AAC Synergy, which will be more resistant to Fusarium Head Blight and Dawn resistance than AAC Synergy.
There's a lot of science in that clip, so let's slow down and focus on the key idea, gene expression noise. Gene expression noise is the small, natural variation in how strongly a gene is turned on or off from one plant to the next. Even when those plants share the same DNA and are growing the same way. It's the difference between identical dimmer switches set just a little differently. That matters because those tiny shifts in gene activity can create real differences in phenotype or how a plant expresses those genes, including how much Fusarium a plant supports and how much Dawn ends up in the grain. Brar's insight was to treat that noise as useful information, a signal pointing to genes worth finding. Another useful term you'll need to understand is SNP, SNP, which stands for single nucleotide polymorphism. Think of DNA as a very long instruction manual written in a 4-letter alphabet, A, T, G, and C. A SNP is a spot where one letter differs from what you'd normally expect. Depending on where that change sits in the code, it can alter how a gene functions. And as Gurcharn explains, some of those changes matter when it comes to how a plant responds to FHB. He grew 200 plants of AAC synergy under identical, tightly controlled conditions, and then looked for differences. Here's what he found.
So I want to circle back a little bit. You talked about the project looking at Fusarium Head Blight in AAC synergy, and that and you used the term gene expression noise. So the genetics of a plant, that's inside, that's how it's coded, and the phenotype is how it expresses those genes. So the plant that you see.
Yes.
And within a specific variety, we would expect that how it appears to be consistent. And then we have a pedigreed seed system that is reliant on that visual appearance being steady. A variety is considered distinct, uniform, and stable, so it's separate from other varieties, and it's the same for all plants, and it doesn't change over generations. How does gene expression noise fit into that?
Sure. So, for this study, we used the breeder seed source, and breeder seed is the purest form of seed. So even though we say that, okay, if you have, 1,000 plants of AAC synergy, which might be F6 derived, I believe. So at F6, you already have like 99 point something homozygosity at all these base pairs, genes.
Yes,
And
they're all the same.
all the same. However, it is only theoretically possible. Practically, it's never possible. So there are still a handful of genes where you might detect some SNP variation, right? And that's what we try to capture. And some of these SNPs are in the genes that
There's what
regulate
SNP is a single nucleotide
polymorphism.
What is that?
So it's basically our DNA is coded in the language of 4 letters, A, T, G, C,
Yeah.
nucleotide bases. So for a specific, that chain of nucleotide bases basically makes a gene, and then that gene translates and makes a protein. And then protein can either play a structural role or a functional role in any living organism. Anytime a nucleotide base is changed, it changes the amino acid, and change in amino acid can lead to change in protein. So when the protein is changed, then the structure or function could be different. There are also some SNP mutations which do not change the structure or function, but so in this particular case, when we took AAC synergy from breeder seed, we grew plant from single seed. We grew a plant, then the seed we got from that plant is what we used for the experiment. And then when we sequenced every single plant, we did not find many SNPs as expected because it should be fairly homozygous.
Okay.
But we still identified a couple thousand SNPs. Now, just for context, the genome size of Barley is 5 GB, so 5 gigabases. So a few thousand SNPs is actually a very negligible number of SNPs.
Fun fact, barley has a genome size of over 5 gigabases, or GBs, which is a lot compared to the 3 gigabases of the human genome. To translate for the rest of the field, what Gurcharn's team found is that within one variety, there are individual plants that are naturally doing a better job of resisting FHB and limiting DON accumulation. Not because they're a different variety, they're not, but because of subtle genetic variation that can now be identified and tracked within the molecular markers. Those markers are tools that breeders can use like a GPS signal inside the genome. Instead of waiting to see how a plant performs in the field, they check for the presence of a favorable gene early in the breeding process and select for it. It's upstream research, the kind that doesn't put a new variety in your bin next year, but it shapes what's available to you a decade or more from now. That's a thread we'll pick up in episode 6 when we look at the full pipeline from lab to field. 4 segments of research, all of it pointed at the same problem. Kelly Turkington gave us the framework, weather, variety, rotation, which ties directly to disease presence. 3 factors. Get those right first before looking to a fungicide pass to solve the problem. And when growers who were doing everything right were still getting hammered, the research community got to work on why. That led to 2 practical improvements. Keeping your herbicide pass separate from your fungicide application, make sure your fungicide application is targeted and useful, preserving the tool for when it is most needed. And understanding that the ideal spray window does not perfectly match what the label states, allowing farmers to be more precise in their timing and more effective with their fungicide application. Randy Kutcher reminded us that Dawn isn't the whole story. Fusarium produces a range of mycotoxins, including some that can evade our current detection methods, and that European markets are already paying closer attention. And Gurcharn Brar's work on gene expression noise opens a genuinely new door, the possibility of selecting better performing plants from within varieties we already grow, using tools that are only now becoming available. FHB is still enemy number 1, but the research is gaining ground. Next episode, we're shifting our focus to the diseases that don't always make the news. Bacterial leaf streak, scald, net blotch, spot blotch. And what's happening with fungicide resistance on the other side of the world that should have every barley producer paying attention.
Thanks for listening to the BarleyBin. Rate our podcast, subscribe, and tell a friend to tune in wherever you listen to podcasts. Brought to you by SaskBarley, because it's time to get in the bin and get itchy.
Produced at
Sound Lounge by T-Bone.